공감상 안에 당 안조 biomolecules ki ane topic enzyme lai re ane enzyme lai ding anjo karla wala na enzyme is a biomolecules re ane compound chire enzyme the ane enzymes the proteins almost all enzymes are protein. So enzyme the kachila na enzyme catalyze the reaction rate of reaction increase the rate of reaction biochemical reaction the is slower that the speed up changing the compound dila kalena chemical dila enzymes there. So almost all enzymes are protein. Enzyme mang che karelana proteins. An enzyme which is not a proteins is ribozyme. Ribozyme is an enzyme which is not a protein, it's a nucleic acid. Enzyme shambat sangma kare proteins. All enzymes are protein, but all proteins are not enzyme. Ra enzyme sangma protein re and protein sangma enzyme ba khepki mane. Ta protein member ki enzyme karana ribozyme ra ane ta nishin ki ta enzyme la karana enzyme structurally enzymes have crevices or pockets called active site substrate ngan bind chese ki ane site chure dila kalere po active site the active site ke nang la kari fit chire substrate fits into active site substrate chura ani reaction and doing substrate the enzyme ngal bind chura and active site la chugin ki reaction now doing the and the substance the color substrate so substrate fits into active site and enzymes are highly specific in their action ra enzyme the kachilana substrate chigla machine and shemba substrate jumar they are very specific active site the korang the sobda chikpa substrate kari jum is tep jumar so they are very specific in their action. Enzymes speed up chemical reaction la biochemical reaction the very slow reaction so enzymes can enhance the rate of reaction. So now reaction the increase share la enzyme kachu gugre la jora they requires in a minute amount they are required in very little amount pe nyu kon dur mangbu shu ni de pyen chu ko ma nyu chu na dur enzymes are recycled at end of biochemical reaction and chemical reaction biochemical reaction chito cha shula enzyme the reaction yam da utilize chitu ma enzyme ko range go ne ka chingure yange ko ne peju che yure. So now it is the enzyme reaction siddhant. Substrate tangi the substrate ngonso karilin yure sucrose the kare disaccharide wa glucose and fructose ki sukyo bound together glucose and fructose ki sukyo the compound the substrate reda that the enzymes and enzyme the substrate jarsala active site yur la sa this substrate the jarsala active site the enzyme the active site. So enzyme substrate binds to the active site form an enzyme substrate complex. That the substrate raw, the active site raw, the active site the active site is the substrate the binds in a karchare enzyme substrate complex char enzyme substrate complex shaktu kabla and the active site la now g take your fit chig substrate the enzyme binding the substrate and enzyme place stress on the glucose fructose bonds and bond breaks and dila kharchiglan this substrate kunan judo dila kharchiglana enzyme ki stress chene minde kharchiglan bond breakdown chene kharchigre substrate the kharchig reaction to substrate the kharchagure substrate the product chagure so glucose tang fructose ni bond breakdown chene glucose factory law ni tesa so this is a product so substrate ni product ni tesa substrate ni product ni tesa jula enzyme ni ange korang go ni recycle here so products are released and enzymes are free to bind other substrate the enzyme de kachere korang free chatur substrate shimbo bind chhes chatur really so this is the action of enzyme. And now next role of enzymes in biochemicals. The role of enzyme in biochemical reaction. So enzyme की biochemical reaction ना. So biochemical reaction ना नंगे देश क्या chemical reactions in living organism, respiration, photosynthesis करने ना metabolism सांगमा. अंजुस living organism ना chemical reaction युंके सांगमा biochemical reaction. So दी ना enzyme की role खरी युरे enzyme की role करे रे enzyme enhance the rate of reaction. Suppose example del reaction steer. Carbon dioxide reacts with water to form carbonic acid. Di niche reaction ne product di to la enzyme su grow chere carbonic anhydrase chere. Ta dele yeast the absence of in the absence of enzyme 200 molecules of carbonic acid forms per hour. किसी absent the enzyme the mena carbonic anhydrase la enzyme the mena and carbonic acid means choosing na carbonic acid ni kya machting marel reaction them. In the presence of carbonic anhydrase 6 lakh molecules of carbonic acid forms every second. Second ching na enzyme di yola ta name in there. And second ching na la ane kaje carbonic acid 6 lakh ne thugre. That means kari kya orkus. Di niche reaction niche dekha je ko reaction ne to ya carbonic acid ngor. Ina kharche the enzyme yo me ke kari kya par kyu song mangyung khache ten. That means rate of reaction, speed of reaction. किसी enzyme यो ना reaction speed छ reaction कच्चे speed छ ने मांग जो तुंगरो product मांग तुंगरो so देला product मांग तुंगरो enzyme में ना कच्चे तो reaction दिपे slow यो तो कल चुजी चिंग ना ला product दिगम संचित तो 200 में चिंत हो मारे. So now comes to the how do enzyme brings about such high rate of chemical conversion. So enzyme की ता देखा लगा enzyme की high rate of chemical conversion chero substrate ni product gyurech to and the speed the go condense in sugo rel the go condense sugo lana ta the graph dita the x axis re the y axis the progression of reaction gyogle lago the got energy required substrate the de do substrate the अनि कुरा एनर्जी ची राजने सबस्ट्रेक्ट द एनर्जी ची सबस्ट्रेक्ट द पोटेंशियल एनर्जी ची राजने सबस्ट्रेक्ट द कार्य करे ट्रांजिशन स्टेट ची रोड इंटरमीडिएट स्टेट र अनस्टेबल स्टेट ची रोड अनस्टेबल स्टेट ची ट्रांजिशन स्टेट ची रोने अनि देखा ची करे प्रोडक्ट छकोर सो प्रोडक्ट छतु कला सबस्ट्रेक्ट द स्टेज रो इंटरमीडिएट स्टेज ल दिदु कप्ता दिस इंटरमीडिएट स्टेज दिदु कप्ता सबसेक्वेंट द लेवल यो तो dingi parta la energy difference suorta energy difference sokyo di kadle kebar gyu rore with enzyme without enzymes kichi enzyme mena substrate ta transition state parchi gora substrate ki transition state intermediate state gora kuni part energy difference digam sur kichi enzyme look bina kuni parta churwa substrate ta transition state by energy difference digam sur so enzyme yana karchu energy difference or transition state and substrate by energy difference the nyunga do and there substrate maluna kharchi do kare enzyme me na energy difference the ma do so the energy difference la kalegre activation energy is like so activation energy the activate che substrate the activate che la energy choro activation energy required choro the energy required the kare enzyme yona nyunga yore enzyme me na energy difference the ma yore energy different activation energy is energy different between transition state and substrate down transition state पर energy difference हो रहा है activation energy ले activation energy the enzyme यो बता मैं क्या पर यो रेला enzyme यो ना activation energy न्यूमा यो रे enzyme में activation energy महा so now enzyme यो ना खर्ची उरला ना substrate the product लग यो रेला energy न्यूमा रोड enzyme में ना substrate the transition state के अंदर जिन्हें product लग जो रेला energy महा रोड रेला So activation energy is the energy difference between substrate and the transition state. That is activation energy ko the exothermic reaction. Exothermic reaction lana kharji lana exo means outside thermic means release heat release reaction delay. If the energy level of product is lower than that of the substrate दिखान दिलाओ product की energy level the substrate level le ma yuna de ma do and didu kala kharchi la ni di reaction kaptula energy kharchi le reaction kala heat release so this is called activation this is called exothermic reaction endothermic reaction la energy requiring reaction if energy level of product is higher than that of the substrate product energy level the substrate la tho tu kala so energy lengor the reaction dil kala na endothermic reaction. So substrate has to go through much higher energy core transition state before transforming into product. Dite jila sa substrate the product machang ene kaudore energy level tho je ruko. So energy level tho rogyo dela energy level tho dela kala transition state. Ko transition state the drone shine product sa machine kaptu product cha tu bo. So now activation energy is a difference in average energy content of S from that of the transition state. Transition state tang substrate content ni patala energy difference or average energy difference called activation energy. Enzyme eventually bring down this energy barrier, making the transition state of substrate to the product more easily. The activation energy digi kharchiglan activation energy mara the enzyme digi kharchiglan energy difference to the nyunga sugrela graph dinando activation energy the enzyme yone activation energy nyunga suda energy difference energy barrier the nyunga sugrela nyunga sudu kabla ane kharchidu lana max transaction of substrate to the product easily. Substrate the product like urea the yoga urea. Energy nyunga sutu kabla and substrate the product urea the lela chegre. So enzyme yona the energy barrier the is bring down nyunga su. So now comes to the nature of enzyme action. Enzyme action tejin khachi the chikuchi kyar. This substrate the enzyme active site. Substrate bind to enzyme active site and active site is altered reaching the transition state enzyme substrate complex. Kuzuda substrate the active site la bind chigri and active site is change chigri reaching the transition stage enzyme substrate complex site. Due to binding the bond in substrate molecules are stressed and become less unstable sheta mere ane substrate molecules bind chigyo the stress ma less stable into kabla ane khunsu ki ka stress in circuit chinne khacha dure bond the breaks ane enzyme substrate product complex है product complex है तो का जन binding lower the overall energy level of transition state activation energy reaction is then become reduced sasa activation energy nyunga sugre activation energy nyunga sudu kala ane ko ka jere di substrate di active site the enzyme the substrate the chinne khachiro products product complex asa enzyme product complex asa enzyme product complex the product the machro tendu so kaje enzyme products living active site enzyme the and product the khachire enzyme active site shana machlo ten so the products are enzyme the gone pichu ke char. So now comes to the factor affecting enzyme की action जो रहा enzyme की activity ला अनि factor खरे की अनि शुकियन टिगरे ला अनि enzyme लेगा खर्च चे मजे दी अनि factor कवर रहा लेना substrate concentration reaction वाला substrate खर्च amount खर्च ये मिला रहा ले enzyme खुरांग की concentration ला रहा ले and temperature से शिला रहा है temperature high low शोरा all enzymes act at optimum temperature increase in enzyme activity increase but if it keeps on increasing the temperature and enzymes undergoes denature because all enzymes are protein so protein into denature and enzyme activity may and also pH value and pH value also affects the enzyme activity and all enzymes works at the normal or optimum pH. And chemical denaturation lana chemically young on the enzyme denature because enzymes are protein and amino acid bond breakdown peptide bond breakdown so all these factors affect the enzyme activity.